Okay, so let's start. So welcome everyone to our monthly seminar series organized by this ESEP funding special topic network called Integration of Speciation Research. So more information can be found on this network website, and from there, we remind that people can also sign up to the network mailing list to receive automatically the program of the seminar series as well as other news. So today we would be starting a new series of 3 seminar sessions on the genomics of speciation. And with today a first session more specifically on the genomic approaches to identifying barrier loci, with Christelle Fraïsse and Stuart Baird as speakers. So just let me remind you how the session will be organized. So each speaker will talk for about 20 minutes, leaving a few minutes after each talk for specific questions to each speaker. And then we will start a more general discussion session for half an hour. We stay on the same webinar link. So if you have any questions or comments after the talks or during the discussion session, you can write them in the chat. And so it's now our great pleasure to welcome Christelle Fraïsse as our first speaker. So Christelle is a CNRS researcher based at the University of Lille in the north of France. She did her PhD at the Institute of Evolutionary Science in Montpellier with Nicolas Baird before moving to Vienna as a postdoc to work with Beatrice Vicoso and other colleagues from IST, and before obtaining a permanent position a few years later in France. So, so her work on speciation combines really different approaches, theoretical approaches, as well as the development of new statistical methods to infer introgression and barrier loci, and also empirical studies in various types of organisms, from mussels and other marine organisms to also insect and plant species. So she, in her research, she's really addressing a wide range of questions related to speciation, and among which, like the role of sex chromosomes in speciation, also the impact of ploidy on the evolution of reproductive barriers, and also, for example, how incompatibilities accumulate between species and their effects on neutral gene flow. And so today, Christelle will talk about what can modeling demographic history tell us about species barriers. And so please, Christelle, whenever you are ready, and looking forward to listening to you.
Thanks a lot, Carol, for the presentation. I think I cannot show the screen. Okay, no, I can.
I just removed.
It was okay. Perfect. So thanks again for the invitation, really. So I wanted really to talk more broadly first about what can modeling demographic history can tell us about barrier loci in general, and then present a bit a method that we published like 2 years ago with Camille, and then look at some applications. So very broadly, first, I think it's important to ask what is genomic architecture of reproductive isolation or of speciation more generally. And so very simply, we can describe genomic architecture with the number of selected loci that are involved. So from here you have a fertile in blue and sterile in yellow. And so we can have, for example, a very strong single factor along the genome that causes sterility, but you can also have more polygenic model where you need to have a very important number of factors that will contribute to an additive trait. And in between, you have more multi-locus model with a number of factors, can be 2, 3, 4, 5, etc. And that can have quite complicated interactions between them, like DMIs, to contribute to sterility. Then you also have simply the nature of selection that act against hybrids that can also be quite different. So you can have a single locus. In that case, you have underdominance with the heterozygous individuals that suffer a lower fitness compared to the homozygous individuals. You also have the classic Dobson-Schindler incompatibilities, where you have incompatibility between alleles at different loci. For example, here, small a, big b that are incompatible. And so it causes a decrease in fitness. And the fact with that kind of DMIs is that you can easily evolve from one fitness peak to the other. So from one species to another one without crossing fitness value. But at the same time, if you have second in contact with that kind of incompatibilities, they will not act as strong barriers to gene flow. Because in that case, some recombinants are fit, and you can then reconstruct the path between the two parental species. In contrast, you can also assume different types of incompatibilities called here pathway incompatibilities. And in that case, the mechanism is more based on co-adapted loci on the same haplotype within species. And so in contrast, recombination will break down this co-adapted loci, like here and behind the blue ones. And so in that case, as recombination breaks down the co-adapted loci, when you have someone in contact with gene flow, you can keep the barriers between the species more easily. But on the other hand, it's more difficult to evolve at the first place this type of barriers, because you need again to cross, like here, a fitness value to go from one species to another. Another category, very classic one of selection against hybrids is simply local adaptation, where you have an allele that is adapted to environmental 1 and the other one adapted to environmental 2. But in that case, the barriers between species can change depending on space and time if the environment change with time and across space. So you can have different expectations actually for the strength of the species barriers with these different types of selection against hybrids. And finally, we can also ask how these barrier loci are distributed across the genome. So here they are in yellow, and so they can be simply randomly distributed, which will be our null model, but they can also be enriched for different reasons in different regions of the genome. For example, because of functional reasons, they can be enriched in protein coding regions or regulatory elements, but they can also be enriched in regions of low recombination, so depending on the parameters of the genome, so like in inversions or in sex chromosomes as well. So as population genetics, we really want to know if we can detect barrier loci and if we can distinguish between the different genetic architecture of reproductive isolation. And so first, the first question to ask, and genetic theories, the genetic theory will help us to ask to that question, what is a barrier loci? So barrier loci can be described as a loci that cause a barrier to gene flow. So it causes a reduction in the effective migration rate locally compared in the genome compared to the migration rate that will be without the effect of the barriers. So if you want the physical migration rate in the long term. And so this reduction in gene flow locally in the genome is simply due to a reduction in the production and in the fitness of the different hybrids, so the early generation hybrids, but mostly the later generation hybrids. So it's both the selection against hybrids and recombination that will shape this pattern along the genome, and so produce this very local barrier along the genome. And so we can quantify the strength of the barrier to gene flow that act locally along the genome, mainly thanks to Nick Barton's theory that he developed in the 80s. So here you have a plot that describes the barrier strength. So ME, the reduction effective migration rate relative to the migration load-based barriers, as a function of the distance of the natural marker to the selected loci, which is here. And so as you can see, first thing is that more you're linked to the selected locus, the more you experience a barrier strength from that selected locus if you're a natural marker. But also in a multi-locus model, so in that case, we assume multiple selected loci, it will be important to consider this coupling coefficient, which is the main parameter that affects integration, which is just defined by the ratio between the selected strengths of the block to recombination, effective recombination. And so when this coupling coefficient is high, this means that the selected locus are very tightly coupled, and so they act as a single block. And as you can see, the barrier strength will be very strong, even if you're quite far away from the selected locus. And at the extreme case, you have, in that case, when theta is low, it's really more the effective selected coefficient of each locus that act, and so the barrier strength is much lower. So from that theory, we can see that it's possible to detect barrier loci by scanning the genome for regions of reduced gene flow. And so for that, we first need gene flow. So lineages currently exchanging genes. This can be in an equilibrium situation like hybrid zones, and Stuart will then talk about that in his talk. Non-equilibrium histories, so this can be sequential contacts, onset migrations, or very complicated history. But sorry, what we want is really to have currently gene flow between the species that are considered, so not onset migration. And then the second point is that we need to quantify the effective migration rate along the genome, so that's ME. And so for that, it's not so simple because many metrics that we use in population genetics can also be impacted by other factors than selection against migrant. And the most typical one is FST, which is impacted by background selection as well. And so we need to consider demography to really be able to correctly interpret this FST measure. Okay, and then if we want to also get an idea of the strength of the barrier and more generally to the strength of reproductive isolation that could be estimated with this ME over M, we need to quantify this migration rate M, which can be a bit hard with only population genetics, because the measures that we get from population genetic data can be impacted by selection against migrants, and so we don't have this M rate without the effect of the barriers, at least if divergence is very high, or the isolation with that act globally along the genome in the first generations is strong. And also another issue that we cannot really use maybe ecological based estimates because it's very short-term estimates, and it won't reflect a long-term migration rate that is average across space and time. So maybe the easiest way would be to quantify migration rate within species in a similar spatial context than the one that we are considering between species. But this may be a bit different question, because here we really want to identify viral loci, so we don't really need maybe this to quantify the strength of the barrier. So the most used method, as I said previously, is to do FST scans, so scan the genome for peaks of FST. So there are a bunch of different studies that have done that in the last 10 years, let's say, or even more. And so what I wanted to illustrate here is that really, if we take the example of this Elicnius era to populations, we can see quite clearly peaks at color pattern loci, so more very strong single effect locus. But this is when you have a high gene flow between the populations. But when you don't have high gene flow, when it's quite restricted, you cannot really see anything basically here. So it's very hard to detect anything. Also, on the other hand, you can have different types of barriers, so DMIs or pathways. And here you have simulations that show you can also expect peak of FST for that type of different barriers. So different things to take from this. So first, that we should expect peak of FST, or to detect a decrease in the effective migration rate, whatever the nature of selection against hybrids. So this is nice because it tells us that we can identify broadly barrier loci, but if you only use population genomic, you won't be able to say anything about the nature of selection. And on the other hand, we really need specific conditions sometimes for high gene flow to be able to detect the best FST peaks. And also best FST, as I said, is impacted by background selection, demography, and also you have a lot of variation just due to randomness of the coalescing process. So this is the reason why we tried to have more suitable approaches, and a very good approach is the one that is provided by the Klein theory. And so again, Stuart will speak more about that, so I will just say briefly that it's a very nice method that is focused on a shorter or medium time scale, like 300 generations, because you need here to have equilibrium at the selected loci. And so you need to have a Klein hybrid zone if you want to have an estimate of the effective migration rate through the various strengths that you can have with the width of the Klein. Okay, and then you have a second set of methods that I will broadly present here, which are DEMO selective inferences. We can call them like that. So we are focused on very simple spatial model, usually 2 DIMs, and then you're looking at more long-term processes, so it's more many thousand generation on the order of the population size. And the interesting thing here is that you don't need to have equilibrium at selected loci, but you can also study non-equilibrium histories. And so at the end, you want to estimate the demographic parameter, the time of splits, the population size of the populations, and the way people have done to identify barrier loci, or to at least take into account selection against migrant. First, a set of methods I've just it's just using demographic inferences to mimic selection. And so in that case, you have estimates of effective population size along the genome and effective migration rate. But selection is not explicitly modeled. It's just captured by its effect on the reduction of migration rate, for example. So this is on the left, and on the right here, you have more complex method that explicitly model selection. And in that case, you can have an estimate of the effective migration rate from selection density and migration rate estimate. So you also estimate, this is very interesting, the selection density, so the number and effect jointly of the selected locus. But the drawback of the method is that you need here a recombination map because it's based on the negative relationship between recombination rate and integration in the case of polygenetic barriers. So I will now more present this first type of method, the demoselective inferences that mimics simply selection, so it's easier to do. But it can still tell us something, I think, about the nature of genomic architecture. So as you can see, the number of, so this is a review that we published this year. And that show that the number of publications that include heterogeneity in migration rate and effective population size increases, has increased a lot in the last year. And the most used methods are this DAGI or moments, which are based on the site frequency spectrum to compute or approximate the likelihood. But you also have here, I think, unpublished method based also on the on likelihoods, but that uses blockwise information along the genome, and deals, so the method that I present shortly after, which uses summary statistics and is based on IBC or machine learning. Okay, so very broadly, this method first tests demography. So in general, you want to compare current isolation versus current gene flow models. You have different flavors of each of them. And in the second step, you can test selection against migrant by comparing two types of model for the migration rate, one which is homogeneous along the genome and one which is heterogeneous along the genome. More precisely, how the effect of barrier loci can be modeled. So most of the studies have modeled it very simply by taking a binomial distribution. So by assume that a fraction p here of the barriers of the loci are barrier loci or linked to barriers. So ME equals 0 or ME is reduced compared to the background migration rate, which is here M, and correspond to 1 minus P, the rest of the loci. But it's a quite crude way to do to model the effect of barrier loci, simply because when you sample the loci on the genome, they will be more or less linked to this barrier loci, and also the barrier loci themselves are more have a selection coefficient which is more or less strong, and so this will produce a lot of heterogeneity in the ME reduction along the genome, and so it can be better to model the effect of barrier loci with a beta or beta binomial distribution, which only uses 2 parameters, and you can then have a variety of distributions with here a peak of barrier loci M equals 0, and here M equals M, so no reduction relative to the background. So it tells you the proportion of loci in each category of M classes. Then the last step that you can do is to explicitly identify loci linked to barriers by testing models. So the idea here is to first estimate the best demographic model from your overall data set. So for example, here it's running contact with heterogeneity in migration rate. And then using this whole genome best estimate, you simply compare 2 models at each locus, one with and one without migration. So it's a way to summarize the migration, effective migration rate along the genome in a binary quantity. It migrates or it doesn't migrate the locus. Okay, and so then how should we interpret this demoselective inferences? Because A first issue is that, of course, we don't have here, we are considering non-equilibrium histories. And so that means you have an infinity of models that can be imagined to be close to reality. And so, of course, we cannot model everything. And so we have to really simplify the models that we want to test according to the hypothesis that we have at the beginning. And so that's why it's very important to, after doing the inferences, have a goodness of fit step. Because if your model is really very far away from the truth, then you will just say wrong things and wrong interpretations. So doing a goodness of fit, so comparing, for example, in the PCA, the prior simulations with the posteriors of the best model and the observed data, which is here in yellow, will tell you if the model that you're considering as the best is good enough for the data. Finally, another complex thing is that we have here a distribution of effective migration rates. And so, as I said before, we can interpret that very simply as saying that m equals 0 are the barriers, m equal m correspond to the background rate, and in between there are loci linked to barriers. But actually, this also depend on the overall migration rate, background migration rate. So if you have a strong reproductive isolation, so if migration rate is small and reproductive isolation that acts genome-wide is high, then you have a strong barrier with that distribution. But on the other hand, if you have M large and so very weak reproductive isolation, this distribution will tell you that you have a weak barrier. So you cannot really interpret, what I want to say is that you cannot really interpret this distribution of ME just like that. You really need to have the background migration rate and other estimates to correctly interpret that. Okay, I'm done with the time, but maybe very quickly, I will try to show you some results if I have maybe 1 or 2 more minutes. So this was just to tell you about the Stills model method that we have that can be used also online. And that we, maybe I will just, yeah, sorry for the time. Maybe I will go to that slide where here we try to see if DILs can accurately identify loci linked to barriers. So what we've done here is that we've simulated 10,000 loci with specific migration rate, so high migration rate or a barrier locus typically. And then we applied this two-step method that identify loci are migrating or not. And so we calculate the proportion of loci which are inferred as barriers with DILs. And as you can see, with very typically with very long time of split. So when the population diverges for a long time and then they just are in contact for a short time with high migration rate, you correctly infer most locus as barriers when their migration is 0, and as locus as non-barriers where migration is very high. But as you can see here, when the time of split is very recent, so the species did not diverge for a long time, you cannot really correctly infer at all which are the barrier loci or not. So it's very important also to know in which condition the method works well or not. And typically it works well when the signal at barrier loci is very different from the rest of the genome. Okay, and then so we applied this machinery to 61 pairs of animal taxa. And so we get the proportion of the introgressed genome as a function of genomic divergence here. And so it quite mimic well this second, this gray zone slide here, where you have for a very lowly divergent population, a high proportion of introgressed genome, but for very divergent species, you have nearly no integration along the genome. However, as I said before, you really need to interpret this P in the context of migration rate, because here, for example, for vast population, which for vast species pair, which are very divergent, you have a high proportion of the genome that is introgressed, but vast maybe just simply because here M is very small. So again, we need to interpret the vast patterns in light of all parameters inferred. Another figure that we had is similar. We have the proportion of loci linked to barriers as a function of FST for like 60 pairs of marine taxa. In that case, you would expect a positive correlation, but it's very weak and mostly contributed by the extreme here of the distribution. And this again tells us that we cannot really summarize the distribution of ME with only a single parameter P and the bimodal distribution, because here all the points were done with a bimodal distribution of ME. And so to finish, because of this issue with Nicola, we wanted to really try to study the genomic distribution of ME, and thus using a complex of meticulous muscle. So this complex of species is very interesting because you have replicates of populations of different species. And so you can have from ear to ear a sort of continuum of reproductive isolation with intraspecific species in the same geography versus at the other extreme interspecific with different geography. And so you can compare the genomic distribution of ME across this gradient, and as P increases from left to right, you also have a very a change in the shape of the distribution with at the end a bimodal distribution there. Okay, so yeah, maybe I will just stop here because I'm a bit late, I guess. So thanks, yeah, thanks a lot. And if you have questions, please let me know.
Thanks a lot, Chris, for this really nice first overview of the methods and also this thoughtful presentation and interesting approach you just described. So if, I don't know if there are any questions in the chat. We have maybe time for one or two questions quickly. I can't see any. Anyone will have a question for Chris? Yeah, Stuart.
Hi, Chris.
Hi.
I'm just interested in the negative binomial distribution of, so that was barrier strength effect. So it's a great distribution because you can do a lot of things with just 2 parameters. But I'm kind of cheating because we've talked about this issue before. When you have that heterogeneity in effects, then the order of the loci along the genome and whether they're clumped or far apart starts to make quite a lot of difference to the outcome. So I guess even though the distribution, the negative binomial distribution is simple, the variance in outcomes must go up quite a lot.
Yeah, I see. So you mean that having a distribution can give you some information, but if you don't have the distribution along the genome, you cannot really interpret things well, because it really depends on how barrier loci are clustered across the genome. Is what you mean? That?
Yes, both clustering and ordering. So you could.
Yeah, so I totally agree that this is something to consider as well, and so I think I didn't have time to show that, but another idea is then to like scan the genome, for example, here cis-curved species for reduced effective migration, and try to identify the barrier loci with this. For example, we did that. And so yes, having that information in complement with the distribution of ME, yeah, would be essential, I guess.
Thank you.
Yeah, welcome.
Thanks. We also had another question from Dave Schuker, but I think we will ask it during the discussion session, because I think it's a quite general one. It is about is it helpful to discriminate between different kinds of barrier loci? For example, reproductive isolation barriers, dispersal barriers, pre-zygotic, post-zygotic ones. And so maybe that's something of a general question that we can ask a bit later. So maybe we should now switch to Stuart talk.
Yeah, great. So yeah, if you don't mind sharing the screen, Stuart, I'll start with a brief introduction. So yeah, it really is my pleasure to introduce Stuart Baird. So for those who don't know Stuart, Stuart did a PhD at the University of Edinburgh with Nick Barton. And he's actually since worked in a number of different locations on a diversity of different organisms. So for example, University of Indiana, University of Queensland, and also spent some time at University of Montpellier, just to name a few. And so he's currently a research scientist at the Institute of Vertebrate Biology at the Czech Academy of Sciences, where he continues to focus on his key interests, which I think include evolutionary biology, spatial genetics, population genetics, bioinformatics, and hybrid zones. I think one of the really interesting and kind of unique features of Stuart's work is that it really tries to bridge theory with data collected from real-world organisms, and often features really highly creative approaches to tackle really tough parameter inference. And so with that, I'll just hand over to Stuart, who's going to tell us about genome polarization for detecting barriers to gene flow. Thanks, Stuart. Oh, you're muted as well, so yeah.
Thanks, Sean. That was very kind. Yes, this is a bridging talk. So I'm going to talk about genome polarization, and I'm going to explain this by, first of all, considering some simulated data. And so I'm going to start off with 100 individuals that are spread evenly along some distance, and the order of those individuals is hidden, and we have a model of allele frequency at sites, in polymorphic sites in those individuals, which is model of the sigmoid climb. So Chris got this right. This part at the top is once again about climbs. So we imagine reading off the position of an individual, pulling a prediction or a model for P. We can then do some weighted random choices with Hardy-Weinberg equilibrium expectations. And so we can get this vector here of what I'll call counts of B. So if P squared is the frequency of BB, 2PQ is the frequency of the heterozygote, Q squared is the frequency of the other homozygote, then this is the B count here, and we can simulate these vectors. Now, I'm hiding, actually, I've gone 2 slides at once. So I'm going to simulate some genomes. The polymorphisms are going to be Hardy-Weinberg equilibrium, and I'm going to hide the process that generated the data. And this is how I hide the process. I hide the order of the individuals along some distance. And then I hide the nature of these B counts, because which allele is capital B is implicit in the count, and I need to hide that. And so I use this constant, which is a polarization, and that tells us which of the polymorphism alleles to count. And so let's just move that. The B count is 0, the polarization is 0. And so we're going to count 0 T's, 2 A's. The B count is 2, polarization is 0. So we're going to count this time 2 T's, and so on. And so what this polarization does is it just tells us the polarity of the polymorphisms that we're going to count to form a genotype. So we use the green things here to hide the model from the data that we generate. This genotype data, anything inside the green boundaries is hidden. Now, of course, we can do this for each individual. So here's another individual. This one has a different position. Its expected allele frequency is 0.8. And so we get a different B count and a different set of genotypes. Now you'll notice that if we just sum all the B counts and divide by 2n, we get a hybrid index based on the B counts. And so for this individual, that hybrid index will tend to 0.8 because each of its sites has expectation 0.8 for P. But if we have this genotype data over here, which is outside of our blinding boundaries, it's rather difficult to know what the hybrid index is, because we don't know phi. We don't know how to translate these states back to counts. So here's another example. This one has loci. And once again, we can calculate hybrid index if we know the B counts, but if we have real data, we can't. And so we can now polarize or define what a polarization is. Polarization is just a binary choice of which alleles to count at each of a list of polymorphic sites. We can represent these choices as a string, binary string, and there are 2 to the power L possible polarization of L sites. So that's a very big number of polarizations for 1,000 sites, which is the simulated data. So I mentioned that once we have data like this, which is what real data looks like, it's actually nicer than real data, it's rather difficult to get through the unknown polarity and infer the process. Well, if we could infer those B counts, we could estimate individuals' hybrid indices and their hidden ordering, but the B counts are blinded because phi is hidden. And so what we're going to do today is we're going to seek phi such that the hybrid index, when you translate the genotypes using phi, is the same thing as the underlying hybrid index of the B counts. So we can visualize that same simulation data. So I've used 3 colors here. One, well, is the B count colors. And this is what hidden data looks like. So we don't know the order of the individuals. And to count things, I've just used a null polarization. So I've just used a coin toss to decide which alleles count at which site. And so there are 100 individuals down the way and 1,000 loci across the way. And sorting the individuals by hybrid index, where you use a coin toss phi to do the counting, reveals very little. Whereas if we calculate the hybrid index using the phi of the simulation, then we get a rather clean picture. We then sort the individuals by hybrid index of the simulation phi. Then what that reveals is the process that gave rise to the data. And another way of summarizing that is that if we put the hybrid index on this axis, then we can have a probability density function for the hybrid index. And we see that if only we knew the simulated phi, we would get a bimodal distribution of types of genotype. And so we would have spread out our data along this axis. But if we don't know the polarization, when we just randomize that, we just, by central limits here in Rayleigh, we just get a spike at a half. So widening the problem, we seek genome-wide phi such that this holds. For rail data, we can construct phi across two sites by using pairwise association, either an intersect-equilibrium or correlation. But it's unclear how to unite such pairs because pairwise association signs are not transitive. And even if that were feasible, that method may not scale to large numbers of sites, irrespective of our measure, because if we have 1 megabase of polymorphic sites in our genotype set, then we have 500 billion comparisons. If we have 2 megabases, then we have 2 trillion comparisons. So this doesn't scale well. Now there's an alternative polarization, alternative family of polarizations, which I'll call reference genome polarizations. And these are approximations to the genome-wide polarization, but I'm going to suggest these are circular, low-powered, and biased. So the phylogenetic approximation has 2 kind of circular parts to it, which we assume that when we're finished, we'll get something bimodal. And then we estimate what we think extreme means along that axis. And then we have the low power part, which is we then sample 2 things that we think are extreme representatives. We collapse their ploidy to haploid sequence, and then get a phi phylogeny, which is just the transpose of the sequence differences. So that's how you decide which alleles to count. Now, of course, population genetic approach. Well, I'll just summarize the power issue there by saying phylogeny ignores standard variation. Population genetic approach, kind of classic thing, still has the 2 circular problems, but now instead of taking 1 individual at each extreme, we just take sets at each extreme. So for example, we take 20 extreme representatives. We don't collapse ploidy to haploid sequence, and we get a polarization suited to hybrids. And that is what's called diagnostic loci. So just bear in mind these reference populations here. I'll just briefly tell you what diagnostic loci are. So there's the reference populations. And this site would be a diagnostic locus were it not for this T here. Because that T means that you can't diagnose the individuals in the reference populations by the state at the site. Either this locus is a poor diagnostic choice, or this blue individual is a poor reference choice. But only loci, never individuals, are discarded in the diagnostic approach. And this introduces Gompert's bias against detecting integration, because what we lose is good loci showing integration into poor reference genomes. There's an asymmetry in what we throw out, so we get a bias. And you'll note, if you think about it, the Gompert's bias also applies to the phylogenetic approximation, because you're doing the same thing just with fewer individuals. So we have a circular standing variation weak, Gompert's bias phylogenetic approximation. With the population genetic approximation, it's also circular. You can get more power, but that interferes with Gompert's bias. And so I'm going to suggest that the use of reference genomes to approximate the genome-wide phi is troubled. So an alternative for an unsupervised labeling for genome-wide phi, if we just remember that phi is just a labeling of which alleles to count. Well, expectation maximization algorithms update unsupervised labelings. And we can easily make an unsupervised labeling of the data with phi null, just using random number generator, or tossing the coin if you wish. And so this is a cartoon of the operation of the EM algorithm that we use. So the key thing is that you can always work out a hybrid index for some data and a phi by counting the data in each individual, conditioned on this way of counting it. And there's a corollary to this, a tight corollary, which is you can always work out a polarization given a hybrid index and the same data by matching data across individuals given the hybrid index. So you run the tight loop corollary iteratively, and what it converges to is a co-estimation of the hybrid index given phi and the phi given the hybrid index based on the data and count and match, which are two operations. So this thing is a co-estimation, and you can do that from any null. I think the operation count is relatively uncontroversial. The operation match we do is we match by maximum likelihood, and that gives us a diagnostic index, hence the name of our unsupervised labeling. We call it diagnostic index expectation maximization. And just as a shorthand, if we know how to count the data, then we can count the hybrid index of the data. And so we have a shorthand for our halting state of the EM, which is just phi dm given data. So this is our output. Now, kind of aesthetic reason that we call these things DI and HI is of our rectangle of data, where sites are down here and individuals along here. Along this marginal, we get estimate of hybrid index given phi dm. And that tells us what process is going on in the genome set. And along this marginal, we get estimates of diagnostic index, and that tells us where process is going on in the genomes. And so this little graph here might be, for example, for one of the concentrations of coupling coefficient that Chris mentioned within an inversion. Unfortunately, convergence of EM algorithms is difficult to prove analytically. It's easy to show for what I've done here that phi dm is phi sim. In other words, we can work backwards from the genotypes only and get the full simulation. Another alternative for seeing how useful this is to look at phi dm when you condition on rail data with a really strong prior. And so that's what we did here. I'm using the same colors, but I have one more color, which is when you cannot describe the site in terms of the two most common polymorphisms. And this is the same diagram as I showed earlier, except the change between purple and green is rather more sharp, and the diagram is curved around in itself to fit in more information, because this is 17 million polymorphic sites. So we grabbed this data on 69 mouse genomes of ENA. Taxonomists call these things species, biological species. We aligned all the data to a long read reference, the lab mouse reference. And even on a laptop, DM converges in about 2 hours. What do we learn? Well, the first signal I'll point out is these blue things. So wild mouse immune gene clusters overmerge onto inbred reference. The second thing is part of our strong prior. We knew that it was the case that Mus musculus domesticus had captured warfarin resistance, which is a rodenticide. They captured it from mispretis. And in that little magnifying glass, there's a 45,000 site long heterozygous block. That's part of our strong prior. We're glad we picked it up. We expected it to be there. What nobody was expecting was these chemosensory gene clusters, which have introgressed in the opposite direction. We've got 7 instances, both homozygous and heterozygous. All of them are greater than 15,000 sites long. And this was not in our prior knowledge of this system. And that's the wrong slide. Maybe it isn't. Okay. Looks like I'm going to be in time. So if I say this really quickly, this is my closing slide. So you suggested that I should link back to the topic. I'm going to quote Mallett's 1995 quote of Wallace's critique. So Mallett says, Wallace presents a very clear interbreeding species definition, then immediately dismissed it. Species are merely those strongly marked races or local forms which, when in contact, do not intermix, and when inhabiting distinct areas are incapable of producing a fertile hybrid offspring. But as the test of hybridity or barrier cannot be applied in one case in 10,000, and even if it could be applied, would prove nothing, since it is founded on an assumption of the very question to be decided, it will be evident that we have no means whatsoever of distinguishing the so-called true species from the several modes of subspecific variation here pointed out, and into which they so often pass by an insensible gradation. So Mallett sums up by saying, Wallace is first saying it's practically impossible to make all the necessary crosses to test genetic incompatibility or barrier strength. And second, since series of speciation involve a reduction in ability or tendency to interbreed barriers, species cannot themselves be defined by interbreeding without confusing cause and effect. And we would add that Wallace's first critique argues only that it is impractical to measure all pairwise barriers for the purpose of construction of a tree-like taxonomy, which is not our goal. Modern speciation studies instead center on the nature and genome locations of barriers to gene flow. And the second, circular reasoning critique does not apply to genome polarization, because detecting a barrier in the absence of any prior taxonomic information, so no references, no reference clues, this is not founded on an assumption of the very questions to be decided. And I'm done. Thank you. Questions?
Yeah, great. Thanks very much, Stuart. Really stimulating talk for sure. Does anyone have any questions for Stuart? Yeah, I can start off with one. So yeah, so really like this method, what you're essentially detecting is groupings, right?
No.
Okay.
And I expected this question, so I'll go
right.
to, hold on a minute. Give me a moment. Right here. There we go. So I will take as a cartoon translation of groupings, the structure algorithm. So there's a couple of simple differences. Structure does not infer states along genomes, although Daniel Falush very cleverly managed to get it to do that in 2015 science paper. From the structure manual, the underlying structure model is not well suited to data from IBD. But this is kind of just skimming at a much larger thing. It's the underlying models, the underlying scenarios are entirely different. So K barriers does not imply K+1 structure clusters. And we can see this by just imagining that, so here's my data again. Imagine that I sampled locally like that. I got one polarization, but three structure clusters. It's not the same thing.
Right, no, I understand, okay.
And, in fact, you
Right.
The kind of the beauty of the genome polarization is that you do not attempt to model what a cluster is.
Right.
At all, in fact. Really, the focus is barriers. And I think that should be our focus. I mean, think of a ring species. So how many clusters is that? How many barriers does it have? And is it relevant to speciation? So I think it is relevant. There's one barrier, its clusters are undefined. So let's focus on barriers.
Right, okay. Does anyone else have any questions?
So I'm still sharing, you can try and guess the other questions I prepared for.
Well, can I ask another one then? So
please do.
What happens if you just happen to have sampled a set of genomes and there's more than one barrier?
So I'll just consider this example here. So Christelle mentioned that a lot of us have worked with or work with the Barton machinery for barrier strength. So the units of barrier strength are distance. And so my example here of dropout with distance, this is a 2-barrier data set. And it's still one polarization axis. And if you were to apply Barton's machinery to each step, if you manage compressing that over your site dropouts, then you would get 2 barrier strengths.
Okay,
Is that okay?
Yeah, no, I understand. Yeah, for sure. Okay, so we do have a question from Dave. So in real life, we often have ordering info. Can you specifically add that in your framework, or is it choice you would use ordering to test model? So I guess the question is, I mean, is that a question about using ordering as prior information or something like that?
So I'm guessing it's using ordering of individuals for spatial analysis.
Yeah.
And I would not use it. And it's because of all the arguments from circularity that apply to the Gompert bias and to Wallace's critique and the very way in which we reason about these things. You don't want to beg the question. So you don't want to stick in information that means you're going to get the answer you want.
Right. So so really you think being as blind, actually, so even though we have prior information, in some ways that's just really biasing our outcome, and we should really try and separate ourselves from that. And maybe, I mean, clearly considering that in terms of interpreting the results is certainly interesting.
Yes.
Yeah.
So that's what you do is you, for example, you polarize your genomes, you estimate the hybrid indices, and then you say, how well does that match distance from the center of the hybrid zone I've been sampling? Or how well do these hybrid indices match the taxonomic notion that mus spretis and mus musculus are different things, right, with a complete biological species barrier? And I guess an important thing is, well, kind of a key thing is, with the structure framework, you can't distinguish K1 from K2. In fact, with the structure framework, you can't distinguish K because it doesn't mix within sampling, it doesn't mix over K. But we have various post hoc, ad hoc things like the Evano method. But the Evano method does not distinguish K1 from K2. It can't. But I think the K1, K2 distinction is really, really important. And if you put in prior information, you risk getting a polarization that says there's some barrier there. But if you put in no prior information and there's no barrier there, then genome polarization will give you this. So give you snow on the television screen, and we can have clearly expressed statistical expectations for the distribution of that snow. So you can test K1 versus K2. So don't put any labels in. Hi, Conrad.
Hi, one related question. Do you get snow on the television screen if you just put one of your mice species in? Is that what you get if you don't have a strong taxonomic prior of sampling two well-separated things?
What a good idea. We should have done that, shouldn't we? So I'm going to answer if we were able to go to the field and sample in a way which were each of our sampling points were equally distant from each of our other sampling points, then yes, I would expect snow. Unfortunately, in any Euclidean universe, you can't sample that way. There is no way of getting all pairwise distances equal. And so I think you'll get a departure from snow that is just about a small departure from snow that's just about the spatial sampling and the fact that isolation by distance is everywhere.
Yeah.
Okay, great. So I think we'll stop the presentation part of the seminar discussion. So just everyone join me in thanking the two speakers for really stimulating talks, and then we'll move into the discussion. So yeah, thanks to both of you. Really fantastic. Okay, great. And so yeah, we do have a few questions that have popped up that will certainly be a feature of this discussion.